Hey folks, it's Dr. Gilchrist here with your Biopsychology lectures for the week. We are going to shift gears now that we've finished up talking about things like stress and health, the limbic system, emotions, and stress. And we're going to close out the semester by talking about aging and neurodegenerative diseases. Now, some of this I do have a little bit of experience with. I occasionally did some work in what we call cognitive aging, which is looking at how things like memory and attention change from young adulthood into older adulthood. I did my master's thesis on some aging-related research. I helped out with the cognitive aging lab and was part of a cognitive aging lab in graduate school. And my postdoc, I also did some work and research looking at aging in older adults. So we are going to talk about some of the broader things that can happen with what we would consider healthy aging. And then later on in some of our lectures, we will talk about what happens in various types of dementia. So that's just to give you kind of a heads up about what we're going to be talking about for about the next 2 weeks or so. All right, so one of the things that is becoming very clear is that looking at things like cognitive aging or looking at what happens to the brain in aging is starting to become an area of increased research. And part of the reason for that is that by and large, as our life expectancy increases, given advancements in technology, medicine, and medical care, generally what we have found is that since 1900 or so, the percentage of older adults in the United States population has very steadily increased over the last 100 years and is predicted to continue growing. So this is based off of census data from 1990 and before. So obviously these later years that have not happened yet are mere projections, but I really want to kind of impress upon you that looking at these variety of different age groups, you can see that they make up a very, very small percentage of the American population very, very early on until about 2050, where people 65 and over, which by 2050 will probably include me or very closely me. I don't really feel like mathing it out right now, but they will occupy at least 1/5 of the population based on current trends. Now, with that comes an increased number of people who will suffer from Alzheimer's disease. So this is by decade in the United States. Again, this is based off of older data. So, of course, 2030, 2040, 2050, and even the 2020 here are probably based on predictions. So one of the things that we're going to talk about later on when we talk about the risk factors associated with Alzheimer's disease, by and large, the biggest risk factor is age. The older that you get, the greater the likelihood that you could potentially suffer from Alzheimer's disease. So because the population is getting older, we can expect to see more and more people develop Alzheimer's disease. And Alzheimer's is the most common age-related neurodegenerative disease. So what you're looking at here are different percentages of age groups. As you can kind of see, the percentage of people 65 to 74 developing Alzheimer's disease, you can see that that's pretty low, and it does stay pretty flat. But as the population gets older, particularly over the age of 85, you can see that Alzheimer's disease becomes more and more common. Now, one thing that I will mention is that by and large, whether one winds up with Alzheimer's disease or not, there are a large number of age-related changes that happen to the central nervous system when there is no disorder. So one of the things that I often get asked about is, why don't you study people with Alzheimer's disease? Well, first of all, given my family history of Alzheimer's disease, I think it would be a very, very difficult thing for me to want to do. I have a grandmother who died from she didn't die from Alzheimer's disease, but I have a grandmother who suffered from Alzheimer's disease until September. I have another grandfather with Alzheimer's disease. It's very, very hard to watch, and I don't think I'd want to study it. In addition to this, what makes this incredibly difficult is that in addition to the changes that you see with Alzheimer's disease, these are compounded on the changes that happen in aging anyway, that happen with healthy aging. And so it can be really, really difficult to tease those kind of things apart. Now, what sort of things happen with age in general, and we're talking healthy aging with no disorders, no strokes, no dementia or anything like that. What kind of things can you expect to happen to your brain as you get older? There are a lot of changes that happen with age. In addition to the central nervous system changes mentioned here, you will experience a loss of bone density, loss of muscle mass. And these, I do want to mention that these changes that we're going to talk about are average changes. One of the things that we find, especially if you look at how people change throughout the lifespan, as people get older, the greater variability in people that you see. So when we start with an infant, infants are very much alike, but as they get older, as they get involved with different environments, different experiences, and so on, you're going to see more and more differences. And that's especially true when we're talking about aging. So there are some older adults that really, in terms of their cognition and in terms of their brain health, they may not look that different from somebody who's younger. Like somebody in maybe their 20s, 30s, or 40s, but somebody else may exhibit a lot of change. And part of the reason that you see this variability is because of a lot of different environmental factors. That can include things like diet, exercise, stress management, and so on. But by and large, here are the things that you can expect to happen to your brain and your central nervous system as you age. Global atrophy, though not very significant, you'll see much more significant atrophy with dementia. With that atrophy will come enlarged ventricles, selective loss of certain types of neurons, selective deterioration of different axons and dendrites, decreases in neurotransmitters, by and large decreased blood flow, and decreased metabolism. Now, I mentioned that aging is partially, how somebody ages is partially due to environment, but genes also do play a role. Aging is likely governed by genetics in addition to these environmental factors. How do we know this? Well, a couple of different things. First of all, monozygotic or identical twins tend to have more similar lifespans than dizygotic twins do, or fraternal twins. And that would also be true if we compared identical twins with ordinary siblings. In fruit flies, we do actually find that a genetic mutation in these flies, known as the Methuselah gene, can actually double the lifespan of those fruit flies. Now, for those who may not know, Methuselah is a person from the Bible. The Judeo-Christian Bible. He was the father of Noah and supposedly lived 900 years or more. And then finally, we're going to talk about this in a little bit. We do know that certain types of genetic defects can produce what are known as progeroid disorders. So that means that it accelerates some of the aspects of aging. Now, even though genetics does play a role in aging, I want to impress this upon you just as I did when I talked about things like mood disorders. There is not a single aging gene, just as there is not a single depression gene. There are probably multiple genes associated with aging, and there are many, many environmental interactions with those genes, like diet, exercise, and so on. So I mentioned those progeroid disorders. One of the most well-known examples of these are what are known as Werner syndrome. So what happens as we age? Generally, in normal aging, chromosomes will basically start to become unstable over time. In particular, we'll talk about this more in a minute. So you have something called a telomere. It's basically a repetitive DNA strand that basically holds chromosomes together. And the more that DNA replicates, the shorter these telomeres become. And eventually what will happen is that because the chromosomes become unstable, because the telomeres shorten, the cells basically stop dividing. And that's why you see some of the things you do with aging. The skin loses its elasticity. The hair becomes gray. And things like that. Now, in somebody who has Werner syndrome, the same thing happens, but this halt in DNA replication basically starts in puberty. So typically people that have this genetic defect, they don't generally live past their 40s. Now, what's interesting is that they show all of the same signs of normal aging. You see atrophy of the brain. You see atrophy of muscles, changes in cognition, loss of bone density. I would also mention that this is a really rare disorder. Now, there are some other genetic defects that will produce progeroid disorders. Werner is the one that looks the most like sped up aging. The others are going to be associated with deformities in the first few years of an infant's life. You might see things like wrinkles, fragile bones, but maybe not necessarily cognitive deficits. So in this regard, out of the different progeroid disorders, Werner's really does look like accelerated aging. So the telomeres really seem to play a role in aging. What you're looking at here is an article from DePinho and colleagues back in 2010. This was published in Nature, which is a leading science journal. And so basically what these researchers did is they basically looked at the telomeres in mice. And so what they did is they took some chromosomes. They basically tagged the telomeres, and again, as I mentioned, part of the reason that aging happens is that with every DNA replication, the telomeres basically shorten and the chromosomes become unstable. So these researchers genetically engineered mice to lack what's called telomerase. So remember, if it ends in A's, it's an enzyme. So telomerase is an enzyme that basically caps the ends of chromosomes with the telomeres and prevents them from fraying. So as I kind of mentioned, in healthy mammals, the telomeres will shorten with each round of cell division, and that shortening is linked to a variety of different age-related disorders. So these mice basically lack the enzyme that produces telomeres. So these telomerase-less mice tended to be prematurely aged. They were infertile. They had small brains, damaged intestines, really poor sense of smell. And so after observing these mice, DePinho and colleagues basically gave them a drug that was designed to stimulate the production of telomerase. And about 4 weeks after giving the mice these drugs, the visible signs of aging had reversed. So as you can kind of see here, by tweaking enzymes that prevent chromosome tips from unraveling, researchers have shown age-related tissue degeneration can be reversed in some mice. So here was the original headline in the journal article. Telomerase reactivation reverses tissue degeneration in aged telomerase deficient mice. So this original title is pretty straightforward. Sounds kind of boring. And here's what some news sources basically did with this article. Study could keep humans forever young. Yeah, I don't think that's what they said. So the news article basically sensationalizes the original source. This is my reminder to you to be critical when you read about science in the news. Go to the original source when you can. This is a pretty huge leap, but I do find this kind of funny. Now, when we talk about the role genetics plays, I already kind of talked about what might happen with healthy aging. If your telomeres are prematurely short, that might lead to accelerated aging. But in the case of other types of genes, they might play a role in whether or not you develop dementia or something like Alzheimer's. In particular, the big gene that everybody likes to focus on, and we'll talk more about this when we talk about Alzheimer's disease in greater detail in a couple of lectures, we're looking at what is called APOE, which is basically apolipoprotein E. And there are 3 common forms of this particular allele. So we have APOE2, which has a 7% frequency in the population. If you have to get an APOE allele, this is the one you really want. It reduces your risk for Alzheimer's disease. If you have E4, that is the one that increases your risk. So if you've ever done anything like 23andMe or Ancestry and you've tried to look at what different types of alleles you have, if they're looking at your Alzheimer's risk, what they're really going to be looking for is to see if you have this E4 variant. It occurs with about 14% frequency and it increases your risk. Finally, we have APOE3. This allele is the most common and it doesn't hurt your risk, but it doesn't reduce your risk, but it doesn't really raise your risk either. So it's basically functionally neutral. Now, at this point, I really want to impress that even though the E4 variant does increase your risk, we do know that it does increase your risk, but that does not necessarily mean that you will wind up with Alzheimer's disease. There are probably many, many cases of people who have the E3 variant, because it is the most common, and are still developing Alzheimer's disease. So again, your genes are not necessarily your destiny. They will only express themselves in certain types of environments. So if you have the E4 variant, it might just mean taking better care of yourself than you might have had to if you had the E3 variant. It might mean doing more puzzles, making sure you're engaged in cardiovascular exercise, eating a healthy diet, and so on. So some additional evidence that we have that this has a really strong genetic link is that well before the symptoms of Alzheimer's manifest themselves, you can see some differences in young adults based on what type of allele they have. So what you're looking at here is the cortical thickness of the entorhinal cortex, which is near the hippocampus. So you can see our E2 variant, that's the one that reduces risk. They've got a pretty high cortical thickness. E3 is our neutral. You can see that there's a lot of overlap between E2 and E3, but what we're really interested in is our E4 people. And there is some overlap with E3, as you can kind of see, but if you look and compare E4 to E2, you can see that the group that is basically the E4 variant, they have a much thinner entorhinal cortex compared to the E2 variant. And you can see that those bars don't really overlap. So what this tells us is that the genes that kind of code for this disorder, they might manifest themselves very, very early on, decades before one actually has those symptoms. But we're going to talk more about Alzheimer's disease and some different things that you can potentially do to cut your risk in a couple of lectures. Right now, I want to talk about the general changes that happen to the central nervous system with aging. So we're going to talk about gross CNS changes. And just as a friendly reminder to you, when I use the term gross, I am not using that like, ew, gross. I mean gross as in you can actually actually see these changes with your eyes. You do not have to look at these under a microscope. They're even visible in an fMRI scan. So generally what you're going to see that happens with aging, you are going to see an overall general cortical atrophy. So here is an example of what a brain looks like in somebody who is young. Here is a brain of somebody who is elderly. And as you can kind of see, the grooves are far more pronounced. So our fissures and our sulci are much more pronounced. They're much deeper looking. And you'll also notice that our raised areas, our gyri, are looking a little less full. So there's this overall reduction in volume. And with that atrophy, you can kind of see this as well from some different scans. So you can see this atrophy in terms of enlarged areas of the subarachnoid space. So here's the brain of a 27-year-old. You can see that the brain tissue is kind of filling up this space quite nicely. You can kind of see that that is getting quite a bit larger in our 87-year-old. With the atrophy, you will also see larger ventricles. So you can kind of see, here is our 27-year-old brain. You can see the size of the ventricles here. Here is the 87-year-old brain. So the volume increases, absolutely, but also the proportion of cerebrospinal fluid relative to both gray matter and white matter increases too. So something to kind of note. We will see reduced volume in certain parts of the brain. That will include areas such as the basal ganglia. Here's our 27-year-old brain. Here's our 87-year-old brain. Reduced hippocampal volume. It's occasionally why sometimes if you go hang out with your grandparents, if you tell them something, they may not remember it a few minutes later, or you will notice sometimes that they have a tendency to repeat themselves quite a bit. That could be because of that reduction in volume here. So you can kind of see, here's the hippocampus in our 27-year-old scan. Here is our 87. You can see that there's much more cerebrospinal fluids in that space, which would normally be filled by the hippocampus. Now, here's something that's really important. So I'm showing you all of these different areas of the brain that have reduced volume, but not all regions are equally affected by aging. Generally, what we tend to find is that our primary cortices, so our primary visual cortex, our primary auditory cortex, our primary motor cortex, and our primary somatosensory cortex tend to be pretty spared. And we're not exactly sure why that is. Is it because of different neurotransmitter compositions, different rates of development? So what's kind of interesting is that the primary areas tend to be the first areas of the brain, like of the cortex, to develop, and they're typically the last to go. So here you're looking at 3 different areas. This is data from RAS back in 2004. When I was in graduate school, because I was part of an aging lab, I heard a lot about this RAS paper. So what you're looking at here is gray matter volume. In areas like the lateral prefrontal cortex, the primary visual cortex, and the hippocampus. And so what you can kind of see with our prefrontal cortex, generally, once the prefrontal cortex is done developing, which is estimated to be in your mid to late 20s, what we do tend to find is that the volume basically declines steadily throughout age. So basically, once it stops growing, once it's done and it's developed, it's all downhill from there. So you can kind of see a very, very smooth decline with age. Notice that relative to our prefrontal cortex, our primary visual cortex has a much flatter line, indicating a lot less age-related decline. And then take a look at our hippocampus. The hippocampus stays pretty steady until about age 40 or 50 or so, and then starts to drop off in terms of volume quite rapidly. The hippocampus definitely changes, but it's pretty variable. It really tends to drop off in the 70s and older, but we're not really sure whether this is due to normal healthy aging or disorders or diseases that initially attack the hippocampus, like Alzheimer's disease. So there's a lot more that we have to learn about this. Now, with respect to the prefrontal cortex in particular, we're going to talk some more about this, but this is an area that is very highly implicated in aging and other types of deficits. So what we tend to find is that up to our 20s or so, the brain weight is increasing. We're getting that arborization, so the dendrites are growing. We're getting more myelin developing. And then once that's done, it's basically a massive drop. What we tend to find is that from the average cortex, there's about a 14% loss. In terms of the frontal cortex, there's actually closer to a 50% loss with age and about 35% from the hippocampus. So there is a disproportionate frontal atrophy that tends to happen with age. So here we see proportion of volume at age 30. So here's 30. And then here's the drop off. Now, all of the other brain areas shown in this chart are other areas of the cortex, but in particular, notice that the frontal cortex has significantly more atrophy and volume loss than other parts of the cortex. I will also mention that when we're talking about atrophy in the frontal lobes, I am not talking about the motor cortex. I'm by and largely talking about the prefrontal cortex and the orbitofrontal cortex, these association areas that are really critical for a lot of complex cognitive activities. So we get some disproportionate frontal atrophy. Now, some areas with age are going to be relatively spared, but I want you to kind of pay attention to the red. So we have areas here, we get some prefrontal atrophy here, and we have areas around the limbic system. Maybe some of this should look a little bit familiar. Generally, we're going to find that the areas in older adults that we see a lot of this atrophy have a lot of overlap to schizophrenia. Now, that does not mean that our older adults are coming in with hallucinations and delusions and so on. That would require an overactive amygdala, which they definitely do not have. But what we do know is that there are a lot of age-related cognitive changes that look really similar to cognitive signs of schizophrenia. Older adults will experience things like reduced problem-solving ability, reduced working memory abilities. They might be more prone to distraction or repetition or things like that. So this brings up a really interesting thing. Your frontal lobe is really one of the last parts of your brain to develop, but it's one of the first regions to actually show atrophy in aging. And we're not really sure why that is. Is it because of different concentrations of neurotransmitters? Is it because of different patterns of blood supply? We don't really know. And so we've got a lot more work to do to figure out why this is. Let's talk about some other things that you might experience in your central nervous system as you age. You might potentially experience some white matter lesions. Again, these are gross central nervous system changes because they are very easily observable. So a white matter lesion is largely going to look like this, these small little white holes. So they basically present themselves as these observable holes in MRI scans. Now, these are quite common in white matter diseases like multiple sclerosis, where the oligodendrocytes are attacked and you get this progressive loss of myelin. But these white matter lesions are also common in aging, especially in those that suffer from high blood pressure. Now, we're not really sure why these white matter lesions show up. It could be due to a loss of myelin or death of myelinated axons like we see in multiple sclerosis, but it could also be due to a loss of blood supply, dead neurons or weaker neurons and synapses that will actually cause the axons to die or shrivel up as well. That dead space is basically going to fill up with fluid, and we get those telltale holes. And we typically link gray matter loss to cognitive impairment, but remember that the white matter in your brain is really critical for making connections to other areas of the brain. It's by and large how our different parts of our brain communicate with each other. So if you have damage to white matter, you are also going to experience some cognitive changes. Now, what causes the atrophy that we see? As we've kind of talked about, we get there are some different structural changes that will affect the connectivity of the brain as well as the function. That can include things like the loss or shrinkage of dendrites, the loss of certain types of synapses, those connections between neurons, or through death of neurons themselves. Now, in particular, it's the shrinkage of dendrites that will lead to the loss of synapses. So I mentioned that there's a phenomenon that happens early on in life called synaptic pruning. So basically, as you're making all of these connections in your brain as you grow up through infancy and childhood, your brain's trying to figure out what connections it needs and which ones it doesn't. So what we tend to find is that connections that are deemed to be unnecessary are basically removed. And this is what's known as synaptic pruning. So synaptic pruning is normal during development from age 2 to adolescence, but then we tend to find that it levels off. But the synaptic pruning starts up again and increases in older age. Where it was beneficial for children and adolescents, it is not beneficial for our older adults. Now, neuronal death can occur, but it's not the primary cause of shrinkage in the brain. Now, one of the things that we do find is that you can actually stain these cells and count them in slices of postmortem brains, and you can see that they change, that there is a loss of neurons, but what you're going to notice more, and the reason you see this atrophy, is because there are fewer synapses and there's less dendritic branching. And these changes could potentially happen because of reduced long-term potentiation, reduced blood flow to the brain, or reduced glucose metabolism. So here you are looking at a few different brain stains of people with different ages. So here we have a sample from a 22-year-old, a 43-year-old, a 91-year-old, and somebody who's 89 and also suffering from Alzheimer's disease. Now, generally, more white space indicates that there are fewer dendrites, and additionally that there are fewer synapses. So you can see over time there is an increase in that white space. Obviously, these are coming from different people, so there might be some variability here, but in general, aging is associated with that increased dendritic shrinkage and synaptic loss. That's especially true for Alzheimer's disease, and we'll talk some more about Alzheimer's disease and the specific effects it has later on. So one of the reasons that these dendrites might shrink and these synapses might reduce in numbers is because there are some changes in blood flow as we age. In particular, we do kind of see that for those that have Alzheimer's disease compared to not having Alzheimer's disease, there is a significant, So that little asterisk means that there's a significant difference between these two. There is a significant change, reduction in cerebral blood flow with age. Now, notice here we've actually got the vasculature that surrounds the brain. Remember that these blood vessels and capillaries and arteries do not actually make contact with the brain, but aging is often associated with decreased blood flow. That's especially true if you have Alzheimer's disease. The brain is getting a lot less blood to it. So, what other changes happen with age? One thing that we know that changes is a change in neurotransmitter concentration. We know that with age, norepinephrine, which is really critical for that fight or flight system, decreases, dopamine decreases. Interestingly enough, serotonin doesn't really decrease until one is significantly older. And you could imagine that because we know about the effects that these neurotransmitters have on behavior, you can imagine that there are a lot of impairments that could result from these changes. That might include things like reduced alertness, reduced energy, less sympathetic arousal, possibly some motor problems, and also concentration. And that could largely be because of dopamine and norepinephrine being reduced in the frontal lobe. One of the things that we will see relating to serotonin specifically is that many people experience a late life depression. It could be linked to a variety of different things, but it could also be due to a potential reduction in serotonin. Here are some other things that we know that do change with age, and most of this comes from animal studies of aging. One of the things that we do find with age is that there is a reduction in the number of NMDA receptors, there's reduced glutamate activity, and with respect to LTP, we actually find that the threshold for long-term potentiation actually increases. What this means is that you will need higher frequency stimulation to actually create long-term potentiation. Additionally, we do find that LTP decays more rapidly the older that you are. And we also know that for long-term depression, whereas the threshold for long-term potentiation is increased, the threshold for long-term depression is decreased. So that means that this is kind of a forgetting mechanism, and this might explain why aging is often associated with higher amounts of memory loss. So here's a quick review, and we are done with this lecture. So generally, here's what you can expect with aging in terms of changes to the central nervous system. There's going to be overall atrophy. The frontal areas are going to be most affected, enlarged ventricles, as well as an increased cerebrospinal fluid ratio, selective loss of neurons, selective deterioration of axons and dendrites, and this is basically the primary cause of that atrophy that we see with age. Selective decreases in neurotransmitters, so decreased dopamine and norepinephrine, stable serotonin until a later age. Decreased blood flow, physical fitness may help with that, and decreased metabolism. And as I kind of mentioned, age-related changes are extremely variable across people. The older that people are, the greater individual differences that you will see. So we're done talking about most of the major brain changes that happen overall with aging without pathology. Now we're going to talk about what happens to things like memory and attention and so on. And we'll talk more about those in the next lecture.